Okay, so now something completely different. I'm going to present, I just give them a bit of background. Yeah, so a bit of simulation methods which we can use to really look at nanoscale structures at sub-nanometer and sub-nanosecond resolution. So the research group I'm leading is called High Fidelity Biomolecular Modeling, and I'm going to talk to you of two things. I use often the term MD, but in this context it doesn't mean a medical doctor. It means these kind of simulations where you can see really molecules moving. And I'm going to talk a lot about NMR. That's another abbreviation I use. That's when you get really atomistic level resolution wiggles from your system, but you really don't know how to interpret them. And when you put them together, that's where the magic happens. So, but before we go there, a bit of motivation. So this year's Nobel Prize in chemistry went in 1/3 to these two guys who developed a method to predict protein structures from knowing the amino acid structure, so the string of amino acids. They have a computational method, which is called the AlphaFold, which allows you to predict what will be the final folded structure of the protein with a very high accuracy. It used to be that you can only do this with scattering, X-ray scattering methods, but now you can actually do it computationally. The point here is that it is for structure prediction. So it will give you a structure, a fixed structure. But of course, we all know we are living beings. We are not fixed in space. We actually do move. And so the question is, how do you go from these fixed structures to motions of molecules? So let's say you have a biomembrane, for example, here on the left. You know actually the molecules in the membrane are moving. They are not fixed. So how do you go from the fixed structure to something that's actually moving? And we know our experience with NMR, we know that there is a lot of information in these NMR signals. So you will get these zoos of peaks, and you know that these zoos of peaks are related to these motions of molecules. The problem is, even if you have a trained eye, you have been working on the field for several decades, it will be impossible for you to deduce from these actual motions. You cannot make the link in your head. So the question is, I know that this is really precise, but how do I go from this to actually being able to visualize this? So we asked the question, wouldn't it be cool if we can take this data, this NMR data, which we know is super precise, and we could somehow create a video, 3D video, that reproduces exactly this data, so that we can use the video to actually reasonably interpret what's happening in these NMR experiments. And that's what we call the out-of-focused MD simulations, because MD simulations, as they come in a package, they will actually not reproduce this data. So I'll give you an example. I don't know if you can see, but there's a molecule drawn there. Maybe you can see it on the other side. So those are actually the chemical structures of the molecules I was showing you wiggling in the previous slide. And if you now look at the NMR data that we have, we can see that for the tails, so on the two panels on the right, you actually get pretty nice matching with the experiment. Experiment is in black and the simulation is in red. You can see the red dots are more or less on top of the black dots. So there you could say that you can really use the simulation to interpret the NMR data, right? However, in the interesting part, which is, or maybe, but depends what you are interested in, but often we are interested in what actually interacts with the cell membrane. So the water-facing part. That's on the left-hand panel here. And you can see there that there is problems, especially in those carbons that are labeled G3, G2, and G1. They are very far off from what we get from experiment. So you don't know how far off the actual movements are, but you know that they're definitely not right. So you cannot use these simulations directly to interpret your NMR experiment. Okay, the problem with MD is that it comes with thousands of parameters, and they are really heavily cross-correlated. So it's not easy that you would just by hand would just tune one parameter a bit, and then that will be fixed. But because of these cross-correlations, if you tune something, you will break something else. So what we turn into, we try to do this automatically using genetic algorithms. So what we do is that we take an existing model, like the red model here, and we stick it into our genetic algorithm, which means that we take the parameters of our MD simulation, we apply a bit of mutations to them, to create different kinds of parameter sets. We let them mate, so to say, to make new parameter sets, babies. and then we keep testing how well do they reproduce the experimental data. And we run this loop. So we create new sets of parameters, we test them, we take the best ones forward, we let them mutate a bit, and keep going through this loop, trying to do what evolution is doing for animals. And then if we manage to converge, then we will go to the validation step where we'll check that actually we didn't break something when we're creating this parameterization. And it turns out that indeed we can do it. So you can see that this is now our focused model. So we didn't need to change the parameters a lot, but we had to change many of them in a very unintuitive way. But we are then able to do this. So now we have a system where we actually do reproduce each one of those NMR parameters. And then we go from this, and we will test against, again, some other data, like scattering data, to make sure that we actually didn't destroy the system. And then what we have created is now we have a simulation and indeed reproduces this. Now we can look at these nice videos, and it will be a robust way of analyzing the NMR data. So to take home, I try to convince you that NMR provides a lot of high-resolution data of biomolecular motions, so not only structures, but also motions. I also showed you that MD simulations don't directly reproduce this data, but you can make it reproduce it. So you can autofocus by slightly tuning the parameters so that you do get this matching. And I would like to argue that when you make this match, this will effectively create you an Ångström resolution microscope. So something that really will give you structures and dynamics at the level of sub-nanometer and sub-nanosecond, which you could then further use to study nanomaterials, interaction of drugs with membranes, interaction of drugs with protein, cryptic sites, and so on. And with that, I was actually super fast. I would like to thank my group, which is the Hi-Fi Biomolecular Modeling group, all my collaborators, and all the people who have funded. And I have plenty of time for questions if you want.